Okay, so today we are going to begin going over chapter 16. We are going to talk about how we go from one cell to become a more complex individual. In other words, we're going to go over development. So we all start as a fertilized egg. So sperm and egg come together, and then you guys are one single solitary cell at that time. After that, you are going to start to develop, and those cells are going to multiply and multiply and multiply. But that doesn't tell us how we become, what we look like today. If our cells just divide, we're just going to be a big clump of cells. We're not going to have hands. We're not going to have legs. We won't have a head, It's just one big mass of cells. So we are going to talk about stem cells. And when they were going over all this stuff, when they were studying it, when they were trying to learn about it, they used model organisms. Reason being is model organisms are ones that are really, really easy to study, and they breed very, very, very quickly. So they used like fruit flies a lot because they multiply very quickly and they don't have a whole lot of anatomy to them. So the nice thing is if they reproduce very quickly, they can change some things in them to see what does what. So here's a picture, guys. This is actually a fly embryo, and this will later develop into a fly. But you guys can see there, it's just like a mass of cells in the middle. So once again, I'm just talking about how do we go from this, and I know this is a fly, but how do we go from this to us, where we have limbs and different organs doing certain things. Okay. So as I already talked about, guys, in the first slide, a fertilized egg gives rise to many different types of cells. We want these cells to, and our key word here is differentiate. Cell differentiation. This is going to be our cells becoming our tissues, our organs, our organ systems, which are going to work together. That's like, if we're talking about the digestive system, that's your stomach and your small intestines working together. But all that goes through cell differentiation. All right. When we talk about morphogenesis, which is the last term here, this is going to help us get our overall shape. This is going to be like the cells getting their shape and then us starting to get the shape of our entire body. So in other words, we are morphing into a human being. So here's a picture from your book, guys. We have fertilized eggs of a frog on the left side. And on the right side, we have an early tadpole. The crazy thing is this picture here, the one on the left and the one on the right, they're actually only 4 days apart. So those cells differentiate so much that they are actually able to form that tadpole only after 4 days. So what I'm saying here, guys, is along with the cells like differentiating, they're reproducing a whole lot. They are going through a whole lot of mitosis to make that tadpole. So if we relate this to human beings, if you ever notice pregnant women, they don't have as much energy as they have during, when they're not pregnant. Why this probably is because their body not only is going through their own mitosis, but that baby inside of them is also going through a whole lot of mitosis, and that requires energy. So the baby is sucking a lot of the nutrients from the mom to use for energy. Okay, so we already talked about morphogenesis, the second one, guys, a little bit. It's just going to be the development of tissues, organs, those types of things. What's nice is once we get I'll just say an organ. Let's get a lung cell. That lung cell is going to divide and divide and divide. This morphogenesis process allows those lung cells to all be identical to one another. That way they have a specific function that they can carry out. And again, if you relate this to last chapter, what's going to be different in those cells? Well, the DNA is going to be identical. However, how that DNA is coiled, when we talk about the chromatin, the heterochromatin and the euchromatin, how what parts of that DNA are going to be heterochromatin, which is not going to be expressed, and euchromatin, which is going to be expressed. That's going to determine what the function of that cell is, because the euchromatin is going to produce certain proteins that other cells will not. All right. So we have these things called cytoplasmic determinants in the egg. So they are all going to be maternal. They're not going to be paternal. In other words, you are going to get these cytoplasmic determinants in your cells from your mom only. Okay? And I got a few examples of cytoplasmic determinants to go over with you. And we're going to go through a bunch of them, guys, but here is the easiest picture I could find to go over cytoplasmic determinants. So we have a zygote over here, all right? Zygote, let's just say it's one cell and that's it. That cell is going to start to reproduce. And as you can see here, it forms 3 different types of cells. So we have one with a pure white branch. We have one down here with a pure green branch. And then we have these ones down here with a green and a white branch, okay? So once these cells get these certain cytoplasmic determinants, these cells here are just going to reproduce, okay? Along with this one here, and this might actually differentiate again, and those cytoplasmic determinants might go to different areas, and they might reproduce. But what I'm saying here, guys, is that we are going to first take those cytoplasmic determinants and make almost like our like schematic cells out of it. Like we're making a schematic or a direction pamphlet, I guess you could say, to go by, which is like that original cell. And then that cell can reproduce, and those cytoplasmic determinants would all be the same. Here's another picture of it, guys. All right. And you can see here we have, let me move myself out of the way here. We have our cytoplasmic determinants here, and they're going to go down and do 2 different cells here. Okay? So there is A and there's B, and there's this thing called induction, and we're going to talk about induction here in a little bit. So I'll go over that. So for induction, we are going to have signal molecules from embryonic cells, and this is going to cause changes in nearby cells. So what this is going to do, this is almost like a proximity kind of thing, and I have a picture of it coming up. But it's a proximity thing where the one cell is like, hey guys, guess what? I'm a lung cell. Do you want to be a lung cell? And they're like, I can't really talk, because cells don't really actually have voices. But the cell's like, oh, it doesn't matter. Here, you're going to be a lung cell. And it gives it that cytoplasmic determinants, and it makes that cell, which originally is a stem cell, into a lung cell. And then that cell reproduces and just keeps reproducing more lung cells. So here is, this was the first one. So if we go back up, this is A here, okay? But if we look at this picture here, this is called asymmetrical regulation. So there's asymmetrical regulation, and then there is induction. So what we have here in our zygote, we have these cytoplasmic determinants, or the transcription factors, either way we could say. They're going to be on one side of our zygote. When that zygote reproduces, they are all going to be in the middle area here. They're all going to be in one cell. So that way, when this bottom cell reproduces, all of those cells that it makes are going to also make copies of those cytoplasmic determinants and put them in all the new cells that are created. So that way, whatever that bottom one is, let's say that's going to be a heart cell, all of those cells that reproduce from that one are going to be heart cells. Let's say the top one here, let's say it's a skin cell. It doesn't have any cytoplasmic determinants, all right, as you can see from the middle there. That's going to reproduce, and it's going to make a whole bunch of skin cells. Here is going to be the induction process. So we get a group of cells, we got another group of cells here, and don't think these cytoplasmic determinants are the same. But what's going to happen is those cells are going to almost like peer pressure the other cells, like, hey, let's do something. We're going to be these types of cells. And they differentiate and they become that type of cell. Now, there's two different types of induction. There's diffusion and there's direct contact. Here's diffusion up here. So in diffusion, we have our cytoplasmic determinants right here in that area and over here. But what they're doing is they are coming out of this cell and they are diffusing over to another cell in close proximity, right? They're not going to go all throughout your whole body, because if this is a heart cell, we would just have a whole mass of heart cells, which we don't want. But they're going to diffuse and go to other cells that are in that proximity. And in turn, all of those cells, you can see it has like a pink outer there, all of those cells are going to become these, whatever they are, green cells. And like I said, I call them heart cells. Another way is through direct contact here. So in direct contact, these cells, and we'll talk about the difference between animal cells and plant cells here, but they can have a bridge almost, and we talked about this in the previous chapter, a bridge between these two cells to transfer these cytoplasmic determinants. So if these are the cytoplasmic determinants, we can have them go through this little bridge and into the other cells, making that other cell become, once again, a green one. Now, if you remember, and sorry, since I'm writing, you can't see what's down there, but if it's an animal cell, that is called a gap junction. That was from a previous chapter. If it is a plant cell, that junction between the two cells is called plasmodesmata. Okay, so those are your two different types of junctions, depending on what type of eukaryotic cell is, depending on if it's a plant cell or an animal cell. Okay, so our next area is going to be on determination. Determination commits a cell irreversibly to its final state. What determination does, guys, is make sure that like a lung cell won't accidentally go and whoops, it's going to become a heart cell. That would be bad, because if we got a lung cell in that area, it's not going to be making the same proteins as a heart cell would. Okay, so let's just go back to last chapter a little bit. What we did, and remember we went from DNA to mRNA to a protein, and those proteins were specific to the type of cell it was. So when we were studying all this stuff a long time ago, we used those proteins that the cell was producing and expressing to determine what type of cell it was. So we had a cell, let's just say we extracted a cell. We were able to see what types of proteins were in there to determine, hey, what is this cell's job? We couldn't use the DNA because all of the DNA doesn't get transcribed, and then all of that mRNA doesn't get translated. So the first evidence of differentiation that we found was in the production of mRNAs for these proteins. We found what mRNAs are going to be in, let me repeat that. We found out what type of mRNAs are going to be in some cells and then not in other cells. So just a few types of different cells that we're going to go over. First one is called embryonic precursor cells. Embryonic precursor cells, these are cells that have the capacity to differentiate into only one specific type of cell. So they're almost like they have these certain genes in them that is going to commit them to become a certain type of cell. The example they give here is a skeletal muscle. Skeletal muscle, these are going to be like your biceps, your triceps, any kind of muscle that is like does your normal body functions that are connected to your bones. Other, like you have cardiac muscle as well, that's muscle in your heart. But anyway, these are going to have a certain gene in them called MyoD. And here's a little picture of it here. We have this MyoD, and it can, I'll move myself out of the way. It can give that muscle a specific signal, and the mRNA specific signal, to produce certain proteins that a muscle needs to contract. One here they mention is myosin. So without that MyoD, that skeletal muscle is not going to be able to do its specific function. It's not going to be able to contract. Oops, there we go. All right, so our next thing is on apoptosis. And I always found apoptosis extremely interesting just because you guys go through apoptosis in your life before you even knew you went through it. But apoptosis is programmed cell death. Okay, so we're killing cells here. Why would we want to kill cells? Well, a few examples your books gives are if the cells are infected by something, if they're damaged, or if they're not functioning correctly anymore. There's one other one that we're going to go over in a minute, one other function or reason we would go through apoptosis. So here, there is on this side, a white blood cell. Now we have three different types of blood cells. We have erythrocytes, which are our red blood cells. We have leukocytes, which are our white blood cells. That's the one function there. They fight infection. And then we have thrombocytes, or platelets. And these are things that if you get a cut, they're going to help to clot your blood so you don't lose all your blood. That would be bad. And then erythrocytes, guys, I think you know what those do. Just to give you a refresher on that. Erythrocytes, they carry oxygen to all the parts of your body because every part of your body needs oxygen. If you remember, why do we need oxygen? It's for the electron transport chain, for cellular respiration, so that oxygen can be the final electron acceptor and make water. In turn, we get a lot of ATP out of that. All right, so we need oxygen everywhere in our bodies to help make ATP. So during apoptosis, we break down the DNA, we break down the organelles, we break down pretty much everything in the cell, and the cell, it says, is fragmented. All right, all of these fragments of the cell, all these different parts are going to be broken up, packaged up into vesicles, and then they're going to be engulfed by another cell that comes by. So in apoptosis, and I know we talked about this word in the past, it was kind of earlier in the year, but we talked about hydrolysis and cytolysis. If you remember, cytolysis was just going to be the explosion of the cell if the cell is hypertonic to the outside hypotonic environment, and that will cause the cell, could cause the cell to get so big that it bursts. For this one here, we do not want that cell to burst. So cytolysis, cell bursting. Hydrolysis, from I think chapter 3, that was breaking something down as well. So lysis, anytime you see lysis, this means to break. All right. So in apoptosis here, we do not want lysis of the cell. So there's no lysis of the cell in apoptosis. Why is there no lysis? Well, if we explode this cell, and it is like a little small explosion, what this can do is it can damage cells that are nearby, and we don't want that. So we don't want lysis. Instead, we fragment the cell into small pieces, we package it into vesicles, and that is best for that cell that is pretty much dying and all the other neighboring cells. I'll be right back, guys. I gotta go do Hall Duty real quick. Okay, sorry about that, guys. So last thing I want to go over with apoptosis is during development, it's kind of crazy. We start off, our hands and our feet start off actually as, instead of having these nice like fingers here, it's almost like a big web. All right, it's everything is solid here. And then as we start to develop, just like this picture here, this isn't a mouse, and I know it's not really green. This is just to show you, to get you to see the picture easily. But there's our like big blob of fingers as they would be. When we start to develop and get our individual fingers, what happens is, no, the fingers do not grow up. Actually, the cells die in between, and that allows us for our fingers to disconnect and us to get our digits. All right, so kind of crazy there. Just a little thing on apoptosis for you. All right, so we'll stop here for the day, guys, and I will see you next time. You guys can start working on your reading guide, and have a good day.